cardiac muscle we are going to discuss. The cardiac muscle fiber is also called as cardiac myocyte. It is also called as cardiac myocyte. Now, this cardiac muscle, it's interesting for us to know that it is having some properties of skeletal muscle, some properties of smooth muscle, and its own individual property, its own individual characteristic feature. Now, what are they? Let us learn. Basically, if you see the cardiac muscle, it is made up of cardiac myocytes that are short, cylindrical, which are showing branching and anastomosis with the neighboring cardiac myocytes. They are short, cylindrical, they show branching. Also anastomosis, that is, they are anastomosing with the neighboring myocytes. So here we are having the cardiac myocyte showing the branches. This is the second, we are just trying to appreciate the branching pattern of the cardiac myocytes. Are you able to understand? Now you see here the cell, it has got single large centrally placed oval nucleus. Now what is it having? Single large centrally placed oval nucleus. That is the feature of smooth muscle. So if you say there is single large centrally placed nucleus, okay, which is the feature of the smooth muscle. Around this nucleus there is perinuclear halo. There is perinuclear halo. What you can see here, that is, the cytoplasm is present in such a style where there is a hollow that is present around the nucleus, and this portion is called perinuclear halo. Okay, in the same way, every single nucleus is going to show the perinuclear halo that is present. Okay, here also having the perinuclear halo. That is, around the nucleus there is a space that is present, what you are calling as perinuclear halo, and here you are having the cytoplasm that is present, which is rich in mitochondria here. Now coming to that property which the cardiac muscle is similar with that of the skeletal muscle is, it shows transverse striations which are present. Okay, so very clearly you can see the transverse striations in case of a cardiac muscle. So this is the feature of skeletal muscle which you can see. So striations, the transverse striations are very clearly seen in case of the cardiac muscle. So you get your actin and myosin filaments giving that A band, I band, anisotropic band which you have learned in skeletal muscle. Those things you can see in the region of the cardiac muscle. Now it has got its own particular feature. Now what is that particular feature? It is showing intercalated discs or intercalary discs, they are also called as. So here if you see the cardiac muscle, it is showing the intercalated discs, the feature of cardiac muscle, that is the intercalated discs, which are also called as intercalary discs. Okay, so what is this intercalated disc? If you see here, you are seeing in the form of a zigzag line that is present between the two adjacent cardiac myocytes. The cell membranes of the two cardiac myocytes, okay, which are adjacent, they are opposed with each other in such a style. You get a step pattern a zigzag pattern. We have made a video on intercalated disc that you can see, okay, and this intercalated disc is responsible for binding the entire cardiac musculature in such a style that it will contract rhythmically. The cardiac myocyte has got a feature that is the rhythmic contraction. Okay, this rhythmic contraction is enabled, in fact it is regulated very beautifully by the pacemaker of the heart. Now you know pacemaker is sinoatrial node, SA node that is present in the right atrium. So that is making the rhythmic contraction when atria are contracting, ventricles are relaxing. Right? So the blood from the atrium goes into the ventricle, and again when ventricles are contracting, the blood is pumped from the right ventricle into the pulmonary trunk, from the left ventricle into the aorta. Okay, so this contraction, the rhythmic contraction, is the beauty of the cardiac muscle. This is enabled, the entire heart is acting like, the cardiac muscle is acting like one single unit. This is because of the intercalated discs or the intercalary discs, which is making the cardiac muscle as a physiological sensation. It is a physiological sensation, you know. It's a physiological sensation. And with the advancing age, as the person is becoming old, there is deposition of lipofuscin around the nucleus. So there is deposition of lipofuscin pigment with increasing age. In the old age people, we are having the lipofuscin pigment present at the region of the nucleus of the cardiac myocyte. And now, in between the cells, you know, you have got connective tissue that is present, and the connective tissue is full of blood vessels. Now the cardiac musculature has got lots of blood vessels that are supplying it. So here you see these are the capillaries that are present in between the branching skeletal, in between the branching cardiac muscles. Now what is the length of a cardiac myocyte? It is about 15 micrometers in the diameter, that is 15 micrometers in diameter, and whereas the length, it is up to 80 micrometers in the length. That is the length of the cardiac myocyte. Between the different cardiac myocytes, you have got the blood vessels and capillaries which are present. So here you have got the capillaries, okay, that are present with some connective tissue adjoining myocytes. They are showing some connective tissue, and here you have got lots of capillaries. These are the blood capillaries that are present, okay, along with the connective tissue that is present in between the cardiac myocytes. So the small capillaries, numerous in number. Definitely it has got a very rich blood supply, the cardiac musculature, because it is going on working, okay, from the womb to the tomb. So it has to keep on beating up. Okay, if heart stops beating, that is called death. Okay, so definitely it will have good blood supply. And the cardiac muscle is similar to that of the red skeletal muscle. The red muscle, you know, skeletal muscle is of two types. One is the red type. So with the red type, it is more in favor with the red type. Okay, thank you. Let us discuss about smooth muscle. Where is it located in the body, or where is it present, and its arrangement and its different names we are going to learn. Now smooth muscle, if you see, the smooth muscle is also called as involuntary muscle because it is supplied by the autonomic nerves, both sympathetic and parasympathetic, and it is not under our control. It is also called as plain muscle or unstriated or non-striated muscle. So it is also called as involuntary muscle as it is not under our control. It is also called as non-striated striated or unstriated muscle. Okay, the unstriated muscle. It is also called as plain muscle. So there are different names for the smooth muscle. So we are going to learn this in the following headings. So where exactly is it present? Its location or its distribution in the human body. Then we are going to go with the microscopic structure. We are going to go with the microscopic structure. Okay? Thank you. Now coming to the location of the smooth muscles, which are also called as involuntary or non-striated or unstriated muscles. It is situated in the walls of the hollow viscera and the hollow tubes present in the body. Are you able to understand what is hollow viscera? That is, you take stomach, okay, intestines, they are all hollow viscera. The urinary bladder, the uterus, the vas deferens, the fallopian tubes, these are all nothing but the hollow visceral organs. And if you are talking about the tubes, arteries and veins, these are the tubes which are hollow. So they are all made up of the smooth muscles. The walls of these are made up of the smooth muscles. So we are going to learn firstly the hollow visceral organs. When we are trying to talk hollow visceral organs, they are nothing but the stomach, and then we have got the urinary bladder, and then you have got the uterus, okay, and then if you are talking about the hollow tubes that are present, what are the tubes? They are the arteries and veins. These are all having the smooth muscles in their walls. Is it all? No. In the skin also you have got smooth muscle in close approximation with the hair follicle, and that is called erector pili. Erector pili muscle, that is the smooth muscle which is present in the skin very close to the hair follicle. That's why you get the goosebumps. Your hair stands up. Why? Because of the contraction of this erector pili muscle. And also in the eyelid it is present, and then it is called Muller's muscle. It is called Muller's muscle. It is also present in males in the scrotum. In the scrotum, you know, there is a muscle called dartas. The dartas muscle of the scrotum is also a smooth muscle. So these are also present in the eye. If you take, they are present in the iris, ciliary body. So in all these areas, the smooth muscle is present. Now you should understand the arrangement of the smooth muscle is different in different systems of the body. For example, if you take the gastrointestinal system, the GIT which you call, the arrangement of smooth muscle is inside inner circular layer, that is, the smooth muscles are arranged circularly in the inner portion, and coming to the outer wall, it is longitudinal. So inner and outer layers, it is arranged. Inner layer is having the circular type of arrangement of the smooth muscles, and outside it is the longitudinally arranged smooth muscles. Now coming to the urinary tract. For example, if you take ureter, exactly this is reversed, wherein you have got inner longitudinal and outer circular muscle coats of smooth muscle. Are you understanding? And if you see stomach, you have got circular, longitudinal and oblique layers of muscle fibers. You know, stomach is compared to grinder, because it is the churning action of the stomach which is breaking down the food into small pieces along with the hydrochloric acid that is present, and that's what you see. Now the chyme from the food is going into the duodenum, okay? Because of the hydrochloric acid, it is becoming acidic chyme. That's why you compare stomach with the grinder, because it is having different layers of muscle fibers that are arranged circularly, longitudinally, obliquely. It is like a grinder where it can churn the food. Churn, C-H-U-R-N, churning of food is done in stomach. Also, if you see uterus, the smooth muscles are arranged in different directions in uterus when you talk about the uterus. And some people they also say in urinary bladder. Both in the urinary bladder and uterus you see the different arrangement of smooth muscles. There is no specific layers, they are just spread in different directions. So now we are going to learn about the microscopic structure of the smooth muscle. Coming to the microscopic study of the smooth muscle, it is spindle shaped or fusiform shape. Now what exactly you understand by spindle shape? Spindle means it is thick at the center and the ends are tapering. That's what we mean when we say spindle shape. If you observe here, see, it is the center where it is showing the bulge, and at the periphery it is narrowed, and in the center you have got a single oval large nucleus. Single oval large nucleus in the center, and here you have got the cytoplasm. And please understand, the cell membrane is called plasma lemma. This cell membrane is called plasma lemma, and the cytoplasm which is present inside the cell is called sarcoplasm. It is called sarcoplasm. And here if you see, you have got some longitudinal striations are seen, but they are very faint due to the presence of actin and myosin. Due to the presence of actin and myosin filaments, you have some striations but very faintly seen. That's why you are calling it as non-striated. You don't have transverse striations. Please listen. You don't have transverse striations in case of smooth muscle. You have some faintly appearing longitudinal striations because of improper or disorder arrangement of the actin myosin filaments. They are not arranged in the order, actin myosin filaments. So you are getting some longitudinal striations. That's why you call the smooth muscle also as non-striated muscle. And here you have the endoplasmic reticulum, you have the Golgi complex, you have got the so many other cell organelles, okay, ribosomes and all are present in the smooth muscle. Please observe, the central thick portion of one smooth muscle is related to the adjoining cells tapering parts. Are you understanding? The central part of one cell is related to the tapering parts of adjoining cell. So one more cell we will draw. So here again it is the central bulging portion wherein you are having, okay, this is the central bulging portion of these two smooth muscles, and look at the adjoining cells. The tapering portions of the adjoining cells are in close contact or connection with the tapering ends of other cells, and they form gap junctions. They form gap junctions. The junction between the cells is called gap junction. There are some small blood vessels that are present in between this, okay, and some connective tissue is present. So you have got the connective tissue with some blood vessels adjoining these cells. If you take the smooth muscles, they undergo slow, sustained, automatic contraction. It is very slow, sustained and automatic contraction. If you see the intestinal wall, peristalsis is the movement. What you have in the intestine? What exactly is peristalsis? The propulsion of the contents forward is called the peristalsis. Now what is the content present in the small intestine? Food. So the food is moving forwards. That is towards from small intestine it will go to the large intestine. Through the gap junctions, when the muscle fibers are getting the impulse, then it is called unitary type. The unitary type of nerve supply. So here what's happening, the nerve impulse is reaching the smooth muscle fibers through the gap junctions. And there is another type called multi-unitary or multi-unit, it is multi-unit type. In this multi-unit type, the nerve supply is directly to the muscle fibers. For example, if you see erector pili muscles or the muscles present in the iris, they are all supplied directly by the nerves. Now what are the nerves which are supplying the smooth muscles? We have already discussed. They are sympathetic and parasympathetic, both come under autonomic nerves. These autonomic nerves are those nerves which are not under our control, involuntary action. So this sympathetic and parasympathetic, both nerves which are having opposite actions are supplying these smooth muscles. So when the nerve is supplying the muscle, it is called multi-unit type, but when the nerve impulse passes through the gap junction, then it is called unitary type. Now here if you see, what is the purpose of any muscle? What is the purpose of any muscle? Contraction. So here the smooth muscle is also contracting. We said it is slow, sustained contraction. So please understand, if you see, this is the shape of the smooth muscle. Here you are having the actin and myosin filaments, okay? And these points, these actin and myosin, they are forming small delicate bundles, and the bundles are attached to the plasma lemma, and these points are called anchoring points. These are called anchoring points. These are also called as focal densities. These are also called as focal densities or the anchoring points. So at this anchoring points, when you say the muscle had contracted, this long spindle shape now will become oval. So it is decreased in the size. So here you are getting the actin and myosin filaments now like this. So here this is how the smooth muscle will look in contracted state, and this is the normal relaxed state of the smooth muscle. Okay, now these are the actin myosin bundles that is present. I hope you have learned something. Thank you. The intercalated discs or intercalary discs, the special feature of the cardiac muscle we are trying to discuss. So when we draw the histological diagram, we see these zigzag lines, okay? These are basically the junctional complexes which are present in between the adjacent cardiac myocytes. That is the cardiac muscle cells which are present side by side. They are having this, in fact they are connected by these intercalated discs. The adjacent cardiac myocytes, they are connected by these intercalated discs. Now please observe, here we have got the cell membrane of one cardiac myocyte. Here we have got the cell membrane of another cardiac myocyte. And here we have got the cytoplasm. This is the dense cytoplasm of one cell. And these are the actin filaments, the ends of the actin filaments, right? So these are the ends of the actin filaments. Are you able to appreciate? In the same way, we have got, this is the cell membrane of another cardiac myocyte. So here is the dense cytoplasm. This dense cytoplasm of this cardiac myocyte is also showing the actin filaments, the ends of the actin filaments. Now, you see here, they are forming tight junctions. They are forming the tight junctions. This is called the tight junction. Okay, where the actin filaments end. The actin filaments, they are ending in this tight junction. So you able to appreciate the tight junction? Then we have got the desmosomes. What you are seeing here is the desmosomes. Now here what you are seeing is nothing but the desmosomes. So these are the desmosomes. Now what are they doing? The desmosomes. The desmosomes, they are linking the fibrils between the adjacent myocytes. See, there are myofibrils. There are myofibrils which are present in the cytoskeleton of the adjacent cells. So these myofibrils or the myofilaments are linked up. So linking up of the myofilaments is done by linking up of the myofilaments that are present in the cytoskeleton of the adjacent cardiac myocytes is done by the desmosomes. And again, these desmosomes, they are forming gap junctions. They are forming the gap junctions. So here you are getting the gap junction. Now again, we have got the cell membrane. Now is the cell membrane, right? Now you have got the dense cytoplasm. The dense cytoplasm. Here you have got the actin filaments, right? These are the actin filaments. What you are able to see. Now here you have got, what do you call them? The gap junctions. So here you call them the gap junctions. Now what are the gap junctions doing? The gap junctions are enabling the electrical continuity between the adjacent cardiac myocytes. They enable electrical continuity. Okay, because of this enabling of the electrical continuity, the entire cardiac musculature is turned into a physiological sensation. It is becoming a physiological sensation. Are you understanding? So you can understand now the intercalated discs or the intercalate discs. They are seen in the form of zigzag lines. They are present between the opposed membranes of the adjacent cardiac myocytes. Here, if you see the cell membranes of the adjacent cardiac myocytes, here is the dense cytoplasm very close to the cell membrane. Okay, this is the dense cytoplasm. In this cytoplasm, we have got the actin filaments. You have seen these are the actin filaments, right? Now, if you see the tight junction and again the desmosome and again the gap junction followed by again the cell membranes with the actin filaments present in the dense cytoplasm. So this junctional complex which is present, that is the tight junction followed by the desmosomes followed by the gap junctions. So these junctional complexes together they make the intercalated disc. Now what is the tight junction? What is the significance of the tight junction? It is that the actin filaments end in the tight junction. Actin filaments they end in the tight junction. And coming to the desmosomes, what is the significance? The desmosomes link up the myofilaments present in the cytoskeleton of the adjacent cardiac myocytes. They link up the myofilaments present in the cytoskeleton of the adjacent cardiac myocytes. And what about the gap junctions? they are responsible for maintenance of the electrical continuity between the myocytes. So the cardiac musculature is turned into a physiological sensation. So this junctional complexes altogether is called the intercalated disc or intercalated disc, the special feature of cardiac muscle. Thank you. Intervertebral discs is a very interesting topic for us to discuss. The intervertebral discs, where are they present? In the vertebral column. In between the vertebrae, now we have learned cervical vertebrae, thoracic vertebrae, lumbar vertebrae, like that. We have got so many vertebrae, and in between these vertebrae, we have got the intervertebral discs. They are nothing but the pads of fibrocartilage or the fibrocartilaginous discs. The fibrocartilaginous discs which are present in between the vertebrae are called the intervertebral discs. They are responsible for giving 1/5 of length to the vertebral column and also the shape. They give both length and shape to the vertebral column. The intervertebral discs in the region of thorax, they are of uniform thickness, whereas the intervertebral discs in the cervical and lumbar region, they are thick in the front. Relatively, they are thicker in the front and little thin at the back. In the cervical and lumbar regions, the intervertebral discs are relatively thicker in the front, and the movement of the intervertebral disc is maximum or it is more in the cervical and lumbar portions of the vertebral column. The movements of the intervertebral disc is more in the cervical and in the lumbar region. Now let us study the structure of the intervertebral disc. The intervertebral disc, it is having in the center nucleus pulposus. In the center of the intervertebral disc, you are having nucleus pulposus, which is a gel-like substance. This is a gel. It is soft and elastic in nature. The nucleus pulposus is soft, gel-like and elastic in nature. As the person is becoming old, the elasticity will reduce. Surrounding this nucleus pulposus, you have got the annulus fibrosus, the part, this is annulus fibrosus. If you see the peripheral portion of, you are having the collagen fibers. You see the collagen fibers. The outermost, these collagen fibers, the outermost portion of the intervertebral disc. If you observe, they are arranged concentrically, okay? They form lamellae, and these lamellae are showing strong bonds. These are the bonds what you are seeing that are formed by the collagen fibers that are arranged at the periphery, the outermost zone. So between the nucleus pulposus and the collagen fibers, you get, this is the fibrocartilage that is present. These are also very thick and dense. They are acting like shock absorbers. Especially if you see the nucleus pulposus, it acts like a hydraulic shock absorber. It is acting like a hydraulic shock absorber when it is subjected under tension. So under tension, the nucleus pulposus acts as a hydraulic shock absorber. So thank you.